じゃあ、時間になりましたので、特別セミナーを始めたいと思います。今日はですね、MIT のイエーニッシュ先生、MIT も僕が細かいことを説明しなくてもいいと思うんですけれども、今回は、実は僕が前ハワイ大学に留学してた時に、それからMIT とイエーニッシュだと後で来ると思うんですけども、ケビン・エガンが学生の時に、コラボレーションずっとしてまして、それからのちょっとした知り合いなんですけど、今回内藤カンファレンスというのが、湘南国際村で開かれて、それが今日終わって、それで、車で公園に連れてきてました。で、今日はね、とても忙しいんですけども、明日の朝帰るんですけども、その短い時間を縫って生育に来てもらいまして、で、まあ多分話す内容としてはたくさんありすぎるんですけども、今回は、大体中ぐらいのビギナー向けじゃなくて、トッププロ向けで中ぐらいのところをちょっと用意してもらって、話してもらうようにします。じゃあそれではもう時間の方ももったいないので、それでは始めていきましょう。
I'm sorry I can't really talk in Japanese, so I'm afraid I have to talk in English. But anyway, thank you very much for whatever you said. Wasn't probably true. So what I'd like to do is to really talk about things we are interested in, stem cells, and really pluripotency and the potential for eventually for medicine. So clearly the medical challenges which we have today are these degenerative diseases such as Alzheimer's and Parkinson's. And one of the potential solutions for this would be what's called tissue repair or regeneration therapy. But the problem with any transplantation therapy is finding suitable donors, as you know. That's really the big issue. So the promise of embryonic stem cells is, of course, tissue engineering. So, as you know, from embryonic stem cells one can generate in culture any cell type of the body. So as you know, stem cells are derived from cleavage stage embryos. They're explanted, and these cells, the inner cell mass cells, are the ones which then grow out when they're cultured under the right conditions to what we call an embryonic stem cell, which is an immortal cell line. And that's what really is behind this. So for those of you who have never don't know what's involved, I have a very simple movie on this, how that's being done. So this would be here a blastocyst, and this is the inner cell mass cells. So what happens is one digests away with antibodies all the outer cells, which are called the trophectoderm, which form the placenta. And then these inner cell mass cells are put in a dish. And if things go well, they will grow, they will multiply. And the key issue now is under the right stimuli, those cells can be induced to, for example, generate liver cells or pancreas cells, blood cells, neurons, or muscle cells. And that works, people learn how to do this. So of course, principally, this is a source of cells which could be used to transplant a new patient who is suffering of death of his own cells. And people envisage that this technology might be really useful for a number of, for the therapy of a number of diseases. Probably not of all of those here, but for some of those at least. So, what is the goal of stem cell research? I think it is eventually to provide matched cells for customized tissue repair. If you want to think, that would be the final goal. So, it's a question of induction of pluripotency and the quest for eternal youth. Well, that's really an old question in humankind, but it's pretty old one actually. This is an example from a German painter 500 years ago. We envisaged this fountain of youth, where people go on one side in as old and sick, and arise on the other side as rejuvenated and refreshed and young. And surely Lucas Cranach, the German painter 500 years ago, had in mind a pool of stem cells here. This would do this. And actually, when you go on the internet and ask for and put in stem cell therapy, you get 1 million or so entries. And I show you a few of them because it's really important to know how powerful this technology is. First of all, platelet immortality is already on the rise. You see this here from the New York Times article. And stem cell therapy works. You can see here there are already success stories about in the press. But if you go to the internet and look for entries, you saw, for example, human fetal stem therapy, really they treated over 1,000 pages, suffering from many of mankind's most devastating diseases. The future is now, and here's an example of this Mr. Fernandez. You see his picture here, and he says here the picture of mine at 57, and he looks much better than Bill Clinton. Looks like that could be an enormous success of stem cell therapy. Here you have what you can use for all sorts of, this in German now here, for all sorts of great diseases. You can use stem cell, and it's really remarkable what people can do. For example, here you can get the stem cells from an apple, from a Swiss apple. So this is based on the stem cells from a rare Swiss apple, which is an endangered species. So it's amazing what people can do, and but you have to pay for that. So for these stem cell therapies, you easily pay a couple of $1,000s. So there are many of those offers, and you really should be aware of those, and probably pay a couple of $1,000s for those things. Anyway, so therapeutic applications of stem cells, of course, is restricted. So when you get stem cells made from human embryos, which are left over, you can generate therapeutic tissue. But if you would transplant this into a patient, of course it would be rejected, because that would be not immunologically compatible. However, when you use somatic cell nuclear transplantation, as you know, then you would generate a customized therapeutic tissue, so that donor nucleus comes from a patient. And if this was transplanted in a patient, of course it would be not rejected, because these are the patient's own cells. So that's, I think, is a promise. Does this work? And this approach, and you know that nuclear transplantation has been rather controversial in many countries. So a few years ago, we made a proof of principle experiments where we used as a patient a mouse which has an immune deficiency. It doesn't have any peripheral B and T cells due to the mutation in the RAC2 gene. There were 2 steps to the experiment. The first one was nuclear transplantation and the S cell derivation. So we take a skin cell, transplant it, explant it, and generate a cloned blastocyst, explant to get a cloned ES cell, and then simple gene therapy, gene correction was used to correct one of the 2 alleles. And then the second step then, these corrected ES cells were differentiated to blood stem cells and then put back into the patient, into the mouse, they indeed restored the immune system. So it worked in principle. So when you use this now and replace the mouse for a human, of course, this is not an option. Because nuclear transplantation in humans really has not worked. So the real big option was, can we learn how the egg does it? Direct reprogramming. And as you know, of course, Shinya Yamanaka succeeded 2 years ago to exactly do that. So we'll talk really now about this approach. So customized cells for routine tissue here, but it was clear that the only realistic option was achieving reprogramming without the need for human eggs. So the issue was we had to understand the reprogramming rules. How does the egg reprogram the somatic nucleus? So, of course, we can realize the egg doesn't accomplish a miracle, although Dolly looked like a miracle. It's not a miracle. It's a biochemical reaction. So let me remind you that if you know the sequence of a single cell of your genome, you know the sequence of all cells. But that doesn't help you because genes are packaged into chromatin, into histones, to form as many epigenomes, as we call it, as their cell types. So cell type A, B, and C, and stem cells have very different epigenomes. So the big issue is, can you convert differentiated cell back to the pluripotent embryonic state, or can you convert one cell type to the other? So that is the big issues of what I'm going to talk about, of the field. So there have been two complementing strategies to induce reprogramming in culture. One was to understand the molecular circuitry of embryonic stem cells. So what distinguishes a pluripotent cell from a differentiated cell, from a committed cell? And then forced expression of these key genes. So let me briefly summarize what we have learned over the last few years about the molecular circuitry. So what we want to understand is, how does a embryonic stem cell self-immune, how does it differentiate, what controls it? And the first glimpse to this came by identifying a circuitry where on top of the list are these three transcription factors, OCT4, NANOG, and SOX2. You will hear more about this. They have two functions. They act as activators of genes which are expressed in embryonic stem cells, and they act as silencers of genes which are not expressed in embryonic stem cells, but will become expressed when the cells are induced to differentiate. So like HOX genes and PAX genes and so on and so forth. All the developmental regulators, very important for development. So this is really has been a very important, many laboratories contributed to this, has been a very major circuitry. Another very important component of this is that these genes interact with each other. So I added the fourth gene, the big pathway. So the OCT4 protein interacts with the OCT4 promoter, SOX2 promoter, NANOG promoter, and the TCF3, this is another transcription factor. And the others do the same. So it's an autoregulatory loop, which is important probably to keep the right level of expression. I will come back to this. That's a very important parameter of embryonic stem cells. So, as you know, the approach pioneered by Yamanaka involved the generation of transgenic mice, which have a marker, and for example, the OCT4 or the NANOG gene. He used a different gene originally, and you all know that. When these cells, of course, are drug sensitive, because the OCT4 blockers or the NANOG blockers is not expressed in fibroblast, so they will die. So then he transduced them with these four factors, OCT4, SOX2, and two oncogenes, Cubic and KLF4, and selected for the rare cells which had turned on these genes, OCT4 and NANOG, depending what you use. And he finds cells which were resistant to drug. And those cells he called induced pluripotent stem cells, or iPS cells, as you also know. Now, the original cells were not really like ES cells. They really would kill an embryo. They had not really activated the endogenous genes, but they were pluripotent. And then pluripotency was really, a year later, after his original discovery, three groups, Yamanaka's group, our group, and Alien's group, really showed that they were identical to embryonic stem cells by many criteria, and this was then repeated by easily with human cells, as you know. So it's a very robust method. So this would be an embryonic stem cell growing on a feeder layer. This would be an OCT4 iPS cells, and this NANOG iPS cells. They're very similar, and they really have very similar morphology, and they express the right markers, albumin phosphatase, SSA1, a surface protein, and NANOG, one of the genes which was not transduced with the virus. So people have done many, many tests of this, us and other groups, and the conclusion is there's really no difference between these iPS cells and embryo-derived ES cells. But the key question is, of these in vitro reprogrammed cells, are they useful for therapy? For example, can they differentiate functional cells in vitro? And they can. You can see here, for example, which easy as you can generate easily beating heart cells in a culture dish. Well, that is not difficult. But more important is, can they make mice? So you're probably aware, you make mice by injecting these iPS cells into normal host blastocysts. You do this really by, you can see this here as one example. You inject those cells into the blastocyst, then they contribute to the inner cell mass, and what they build is a chimeric mouse. The brown cells are derived from the iPS cells, and the black cells from the host blastocyst. And so in those chimeras also transmit this genome to the next generation. So these cells are pluripotent by many, many very stringent criteria. So the amazing thing is that you have cells growing in petri dish, fibroblasts. You take a single cell, transduce the factors, and generate an organism. And this is actually not a chimera, that's an animal, which is an embryo which is derived by what's called tetraploid complementation, which is a trick. I don't want to go into this. But this embryo has no contribution from the host blastocyst. It's really constructed only from these fibroblasts. So they have the potential to form a whole organism with a brain and eyes and so on and so forth. So it's a pretty amazing finding. So the mechanism of reprogramming, what we know is the viruses, the viral genes, are very highly expressed in the infected cells. The endogenous OCT4, SOX2 are not expressed. And in the reprogrammed cells, those genes are turned on, and the viral genes are turned off. So the maintenance of the pluripotent state depends on the activity of these endogenous genes. They get turned on. This autoregulatory loop. So what happens in between? So what we know is the viruses are silenced and the endogenous genes are activated. And we understand the silencing because some enzymes, methyltransferases 3A and 3B, which are not expressed in the fibroblasts, but they get activated during the reprogramming process, inactivate the virus. So the viruses are not needed anymore, they get inactivated. So it's a really nice system. But we would like to know is how long does it take? What is the kinetics? Is there a sequence of markers which come on? Is there a minimal time of virus expression? And can terminally differentiated cells be reprogrammed? So a number of those questions. I will just briefly summarize what we have learned. So a key experimental tool was to really be able to control expression of the viral vectors. So what we generated is doxycycline-inducible lentiviral vectors. So lentivirals are not subject to silencing. So we can introduce into those a trick, and this is here, use a tetracycline operator, which is a DOX responsive element in a virus where these genes are integrated, and you infect cells which carry a GFP in the OCT4 enamel locus. So these fibroblasts are GFP negative, and you ask the question, after infection, can you turn on these locus, and the cells should get green. So in principle, you add a drug to the infected culture and you activate these viruses. If you take the drug away, you inactivate the viruses. So that's a very convenient system then to study really the questions I asked. Kinetics, how long do they have to be expressed, and so on and so forth. So that's really the experimental system. So what have you learned? Well, what we learned when you look for the first 4 weeks after you activate these viruses, so you add really doxycycline, then some of these factors, for example, alkaline phosphatase turns on after a few days, surface proteins like SSE1 or the de novo methyltransferases DNMT3 or telomerase get activated around a week or 1.5 weeks, slowly. Genes, markers which are expressed on fibroblasts, like Phi1, are turned off within 1.5 weeks. It's really a major rearrangement in the cell. And the cells become green after 2 weeks, never before, as measured by activation of the OCT4 enamel locus. And you will have to express the transgenes for something like 10 days. If you express it less, then you will never get an iPS cell, or revert back to the fibroblast stage. So the conclusions are there's a sequential inactivation of somatic and a sequential activation of pluripotency markers, and you have to express the viruses for 10 or 12 days or so. Otherwise, you don't get any iPS cells. So what we believe is happening, and it gives you a very brief summary here, is a scheme. When you infect cells which carry these markers with virus, then in the intermediate stage, they might become neomycin resistant. That's what the original Yamanaka conclusion was. But they're never GFP positive, in case you carry GFP in these loci. You only get, and these are unstable cells. If you take the virus away, they convert back. Only when you get GFP positive cells, then you have stably reprogrammed iPS cells. So what we think is happening is that these endogenous genes, OCT4 and SOX2, are highly methylated. They're really in a very repressed chromatin configuration. And at this intermediate stage, there's partial activation, just enough to give you neoresistance. You don't need many molecules to get neoresistance. So just enough, but that's really unstable. Only once it's fully reprogrammed, chromatin and methylation, then it's a stable situation. So what's happening is these viruses try to interact, or these factors interact with endogenous genes, and they really slowly activate this autoregulatory loop I talked about. And probably that's where the oncogenes are very important. So cells have to divide. Only when DNA replicates, you can reprogram the chromatin or demethylate. And once you have established the autoregulatory loop, then the virus becomes really superfluous, negative inactivated. Then it runs on its own. So some key questions were target cells versus somatic versus terminally differentiated cells. So when Dolly was born, the question was, has Dolly been derived from a somatic stem cell or from the terminally differentiated mammary gland cell, as it was assumed? So the procedure was so inefficient, 1 in 300, it could not be decided. There was no way to determine retrospectively what was the donor nucleus. And then another question is its use of therapy. So let me briefly address some of these questions. So if you think about reprogramming for other cell types than fibroblasts, you have a challenge. One is the efficiency. Only a small fraction of the infected cells become iPS cells. And some cell types, and we will be very interested in immune cells, might be very difficult to infect. Virus infection. iPS cells carry many proviruses, many copies of the virus. I show you this here on the southern blot. Where off the right lane is the control cell, this is the iPS cell. You see many bags, each a provirus. So a lot of 6 or 4 provirus, 5 mycovirus, california, between 10 and 20 provirus are integrated. So you select probably for high copy number, maybe for the right combination of expression, so the right stoichiometry. That's really very important because only a very small fraction of the infected cell carries so many proviruses. So you select for a rare event. So to study the mechanism, which you're interested in studying the mechanism of reprogramming, you really would like to do this without virus infection. So what you would like, ideally you would like to have a reprogrammable mouse. So we made a reprogrammable mouse. So the way this was done is outlined here. So you take again fibroblasts which carry a GFP in the nanox or the orthologous. You infect those with these doxycycline-inducible viruses, as I said before. That we then reprogram, and you eventually get what we call primary iPS cells. So they're perfectly reprogrammed under doxycycline. And you take the doxycycline away and inject these cells into a blastocyst to make a chimera, as I showed you. So you get a chimera. You call this primary iPS-derived chimeras. So the chimeric cells are derived from one of these colonies, which had been an iPS cell. That's the key. So from these chimeras, you can isolate any cell type you want. Maybe isolate intestinal cells, B cells, T cells, fibroblasts, neurons, adrenal cells, keratinocytes, skin cells, whatever you want. You can explant those, then add doxycycline, and ask the question whether they're reprogrammed to what we call secondary iPS cells. The key thing here is you don't need any viral infection. So the issue is, the cells here carry the same combination of proviruses which led to a reprogram in the first place. They were selected. And now this is really working well. So I'll tell you what the efficiency is. So does the system work? Well, when you do that, you, for example, take immune cells, pre or mature B cells, and explant them without doxycycline. There's no virus expression. But after doxycycline, there's robust induction. So the system works. The viruses did not get permanently silenced. They were still activatable by doxycycline. And when you culture those cells, they give rise to iPS cells. And the system is really very reproducible. So you can see this here. In 4 different independent experiments from these secondary mice, you get almost very similar frequency of reprogramming. In contrast to when you do de novo infection with virus, there are 2 orders of magnitude difference between individual experiments. So this is much more homogeneous and much more reproducible. So the secondary fibroblasts are genetically homogeneous. That's very useful for screening for small molecules that could replace a factor. Now, of course, these cells come from a chimera, which have all factors. So how do we make cells which are missing one factor? Well, what you do is you make the chimeras no segregate the pro-viruses after germline transmission. Like all these pro-viruses in there, and so they segregate. If we did this, we took a couple of hundred offspring, we genotyped, very carefully with all that, and what you find is, and I'll show you this as an example, you find some of these offspring which have all 4 genes, OCT4, SOX2, KL4, and MERK. Then you have a whole combination of offspring which is missing 1 factor. Of course, there are many which are missing 2 factors, many which are missing 3 factors. So this one misses KL4, this one misses OCT4, and so on and so forth. So when you give doxycycline to these cells, to those, for example, which carry a copy of each, they are reprogramming immediately. If you give it to those, none of those reprograms. But if you add the missing factor, they're all reprogrammed. So this, we think, is now a great system. Of course, we're using this now to ask the question in a high throughput screen. What small molecule would replace, for example, in this line, the requirement for MERK, on this line, the requirement for OCT4, and so on and so forth. So it's more convenient to do this way than infecting just with viruses, as many people have done. And it works quite well. So we can, from these primary derived iPS cells, we can really reprogram any cell type we have tried eventually, essentially. Then we briefly come to B cells. Because that's an interesting, it's really the question is, can you reprogram terminally differentiated cells? And for B cells, that's a key question. Is it a somatic stem cell, and it was a nuclear transplantation, or a direct reprogram, or is it a terminally differentiated cell? So that we wanted to test. And the immune system is great because you have always doing immune differentiation. You have these markers, these genetic rearrangements. So the early B cells, you have a D to J rearrangement, or V to DJ rearrangement. So you know exactly when you find this in your reprogrammed cells, this is the donor cell. And of course, in the mature B cells, you have the light chain rearranged. So if you have both rearrangement, you know it was a mature B cell which reprogrammed. And you can isolate these cells with markers, and you know all these transcription factors. So, and this was done by Jakob Hanna. So when you did this with immature B cells, it worked immediately. So we just put in the pro or pre-B cells the 4 factors, and you got iPS cells, they form chimeras, contributed to germline. But when you did it with mature B cells, it never worked. So what was missing? Well, lots of things are known about B cell development, particularly about these late transcription factors, PAX5. And what Thomas Graf and Meinrad Busting have shown, if you inhibit PAX5, you really de-differentiate the mature B cells to an earlier stage. And Thomas Graf has shown when you transduce this transcription factor CVP alpha, which inhibits PAX5, you trans-differentiate the B cells to a macrophage. So we thought maybe the B cells are resistant, they don't interact with these 4 factors. You will have to somehow make them change their identity. So we thought, well, just check different factors with a reprogram. So what Jakob did is he put these factors in an additional candidate gene. So you can see when you look for alkaline phosphatase, CVB alpha gave a robust response. So when he cultured those further, they made beautiful iPS cells, which were absolutely by molecular analysis like iPS cells, and they form nice chimeras, and this is a 100% transmitter, and they transmitted to the germline. So that was very satisfying. Of course, we want to know, are they really derived these cells from mature B cells? And for this, you just do look at the rearrangements. There's a PCR analysis, and the red arrow is in these 9 different B iPS lines. They have rearrangements in the heavy chains, productive and unproductive, and in the light chain. It's exactly what you expect from B cells. And then you can also do more. You can ask the question, are there somatic mutations? Somatic mutations occur when a B cell is selected on an antigen, and they do have somatic mutations. So there were clearly B cells which had been engaged in antibody production, which made these iPS cells. So that was what the summary would be. It had the right rearrangements, so it was clear it worked. So it showed that indeed mature B cells can be reprogrammed. But how efficient is that? Well, efficiency in newly infected cells 1 in 10,000, 1 in 500. So that's many laboratories who've done this. In the secondary system, 1 in 30. So 1 in 30 cells, either mature B cells or fibroblasts, can be converted to an iPS cell. So that's pretty efficient. So you shortcut all the virus infection to make it more efficient. So that's what we believe is very useful for studying mechanisms. So nuclear transplantation versus in vitro reprogramming. How similar or different are they? So I said before, we wanted to learn how the egg does reprogramming. Did the Yamanaka approach teach us how the egg does it? Do we know this now? Well, let me give you my view. First of all, when you compare all the different cell types which have been reprogrammed by nuclear transfer, many, many cell types have been. And people like Hidden Kuizuri was involved in a number of these experiments, and Kevin Eggen has been, and many people, and Yamanaka and many other people. So it worked, and I think for iPS cells, it's very similar. Probably all cell types can be reprogrammed by this. So it doesn't mean it, so there's no difference here in the spectrum, I think. So, but I think there are many ways to reprogram a cell. So what you have to convert is the epigenetic state of the somatic cell to one where this autoregulatory loop is activated, as I said. That is a major, that's all what reprogramming is about. So you can do this by nuclear transplantation. And many cell types have been done. It's a very inefficient way, but it's fast. So OCT4 is activated in the 2-cell stage of the cloned embryo. So it's fast, but inefficient. The in vitro reprogramming with the original Yamanaka approach takes weeks, as I told you. Much proliferation, cells have to divide, it's very inefficient. If you leave out milk, it's much less efficient and much slower. And you can do it with other alternative factors. The B cells, you have to add this additional factor. It's pretty efficient, 3%. So all these are highly modified iPS cells that carry the programs. So this is the only way to make reprogrammed cells which are not genetically modified. So for the future, the issue is maybe non-genetically. So some people think about protein transfection, put a protein instead of a gene, not a genetic modification, or screening for chemical compounds, and there are some success already in the literature. So the key conclusion is nuclear transplantation, in vitro reprogramming. I think they're basically very different mechanisms. We didn't learn how the egg does it. So basically you can induce a nucleus, a somatic nucleus, by many ways to end up in this pluripotent state. So nuclear transplantation, the egg does it a different way than the 4 factors from the Yamanaka. So direct reprogramming then is re-establishing the core autoregulatory loop. It's initiated by the 4 factors, multiple cell divisions, and extensive epigenetic resetting. So I think the key is to re-establish this autoregulatory loop, and then all the downstream genes will be correctly activated. So that's the key mechanism, we believe. So you come to human cells. I think people think about 2 applications. Maybe the most important one is to study complex diseases in the test tube. Now, human somatic series has been achieved by a number of groups, but it's really low efficiency. So should we ask, can we generate a drug-inducible secondary system for the human, as I showed for the mouse, because it was very efficient. So the way we did this was to take human primary fibroblasts, infect them again with these doxycycline-inducible viruses, and generate primary human iPS cells. Now those then are differentiated in vitro or injected into the mouse to make a teratoma. And from those we can isolate the genetically homogeneous secondary fibroblasts, as we did with the mouse. Then you can just add doxycycline, ask the question, can you get secondary iPS cells? Right, so it's very similar, except you cannot make chimeras, obviously. So does it work? Well, this takes a long time. So making primary human iPS cells first takes 1 to 2 months. It's a really long time. And then you have to amplify those for another couple of months. So you're talking about 5 months until you have these primary iPS cells in number. Then you want to differentiate those, another, well, 1.5 to 2 months. Then you have those, and then you can give doxycycline, and you get in 4 weeks, you have an iPS cell again. So these are, it does work, but it year. But it works. So you have different donor iPS cells, and they have the right markers, and they're reprogrammed with different efficiencies. And so these examples here, the differentiation we see is between 0.2% and 2%. It's sort of similar to the mouse. It's not really basically different. You can ask now the question, what's the kinetics of reprogramming? And when you do that, you find in the mouse, as I told you, in the mouse, you get this, as I showed you, the marker expression. In the human, you find the relevant surface marker, like TRA160, are activated after 1.5 weeks. That's different from the mouse. NANOC becomes expressed after 1.5 weeks. Now, this is not a GFP, it's really by staining. And the transgene has to be expressed for about 10 days. So I think the conclusion is the kinetics and efficiency of mouse and human somatic cells, when you use the secondary system, it's very similar. Same efficiency and same kinetics. There's no big difference between the two species, as was suggested by the primary infection. So it's probably the difference might be due to really the problem with getting the viral infection reduced and so on and so forth. Now, what you really want to do is to take cells from a patient, generate patient-specific iPS cells or ES cells, and then study them in vitro. So the idea would be, for example, taking making these ALS-specific ES cell or iPS cells from an ALS patient, lateral sclerosis, comparing this with one derived from a normal individual. And the question is, can you see phenotype when you differentiate those from motor neurons and culture tissue? And indeed, there's some success. Kevin has shown that when you use a mouse ALS model, then you do find a phenotype in the motor neurons differentiated in vitro. So that's a very exciting result, because that could allow you then to ask the question, what goes wrong in this in vitro system as compared with the in vivo system? And particularly important would be if you find such a phenotype, you can screen them for small molecules which would either prevent or slow the phenotype you see, degeneration. Now that would be really exciting, because it's a direct, the way to find for therapy. And we have made Parkinson cell lines from a number of patients, from 85 years old to 50 years old, so old, and they have all the right markers. They are formed teratomas. They're exactly what you would expect. Otherwise, I don't want to go into this. And they look, they have all the right markers expressed, so STUA4, SC4, and all these other. And the key question is, can they differentiate to dopaminergic neurons? And in our protocol, they can. Now, there's maybe not, you can't expect to find a phenotype. After all, Parkinson patients can make dopaminergic neurons, they just die of them. So it might be not really to be expected that you find a phenotype easily in vitro. So I think we'll have to get more sophisticated in our approaches to measure differentiation. Maybe you have to stress the cells and see whether we can see such a phenotype in patient-derived cells versus cells derived from normal individual. Now, can you use those for therapy? And that's, of course, is a really key issue. Is that a useful approach? So, again, in mouse, we made we looked at two experimental systems. One is sickle cell anemia and one is Parkinson's. Let me just briefly go through this at the end of the talk. So, for sickle cell anemia, the question is, can you treat such a major disease by genetically identical, but genetically corrected iPS cells? As you know, the cause for sickle cell anemia is globin mutations. So the globin precipitates in the periphery, and then the membrane of the erythrocyte is disrupted, and they block the small capillaries, and it leads to infarcts of the brain, in the spleen, for example, severe anemia, and very severe disease. So it has been made by Tim Tones, a humanized model for sickle cell anemia, where he really eliminated all the most globin genes and replaced those with the human globin genes, either the wild-type alpha globin gene or the beta-sickle mutation gene. And these mice, and they're homozygous for the human beta-sickle cell globin, they're really sickle, as you can see. So they have a very similar phenotype as humans, and therefore have this huge spleen infarcts. So these mice resemble the human disease in a really remarkable way. So we took that such a mouse. You can see this mouse is really sick. It's anemic, it's homozygous for the sickle cell gene. We took a skin fibroblasts from those and then transduced them with the 4 factors and generated genetically identical iPS cells. Now we really were worried about c-Myc. c-Myc is an oncogene which leads to leukemia. It's rare in many, many leukemias in the human and the mouse. That's a gene which doesn't. So we were worried if you do this, if you just give the mice leukemia. So we had modified the Myc so we could excise it with, by recombination. So after it done its job, we just removed it. And then we corrected the defect, so introduced the wild type human globin sequence, and then these genetically corrected iPS cells were then differentiated to blood stem cells, and they were injected into the mouse, and you can see it absolutely cured the mouse. So I will tell you now what the data are for this. So if you are admitted to a hospital and the doctor assumes you have anemia, well, you might measure the hemoglobin. You can see the hemoglobin level in our control mice, in the heterozygotes. At this value, but in the sickle mice, it's less than half. 4 weeks after transplantation, the hemoglobin is almost restored to normal. And another measure would be the hematocrit. Although the doctor would do hematocrit in these mice is very low, and 4 weeks later it's restored to almost normal. And you can look for what type of globin is made in these mice. So this would be a mouse which is homozygous for the wild type alleles, it's only HbA. This would be heterozygous mouse. They have about 20% HbS and 75 or so HbA. It's not 50-50 because there's selection against the erythrocytes which are homozygous for the sickle globin that just slides away. So you have less of those. But of course, the sickle mice have 100% HbS. Of course, what else do they have? And this would be known in 4 weeks or 8 weeks after transplantation. You can see this almost restored. This is the peak of HbA, which of course totally missing here. Almost restored to the control level. Not quite, because these mice are partially irradiated to make space, so there's still some sickle cells in the periphery, so they have a little bit more. But the point is, it's very stable for many months. And this shows you just, again, clinical data here. We're talking about hemoglobin level. It's very low in these mice. The red blood count is very low. They're very anemic. Hematocrit is down. The reticulocyte count, immature blood cells, is very high because bone marrow cranks up immature cells. It's very high, and these animals don't gain weight. But 8 or 12 weeks after transplantation, you see this hemoglobin is almost restored to normal. The red blood count goes up, not quite to the normal, but substantially. Hematocrit goes up. The reticulocyte count goes down, not quite to normal because still the bone marrow is somewhat stimulated, and the mice gain weight and then they get tumors. So I think the conclusion is this approach is really rather realistic for diseases of human bone marrow, or diseases such as sickle cell anemia, thalassemia, Fanconi's anemia. I think we're really much more realistic because we know how to do bone marrow transplantation. I think that's the key thing, delivery. So I think the prediction might be that if people will use the iPS approach for cell transplantation therapy, diseases of the bone marrow might be the first ones to be used. So Parkinson's, the other model, as you know, dopaminergic neurons lead to this movement disorder. And clinical trials have shown that if you have the right cells, and the cells people have used in these clinical trials come from aborted fetuses, from the brains of aborted fetuses, and those are stereotactically injected into the substantia nigra when dopaminergic neurons die, they can restore the movement disability to a remarkable degree. The problem has been that you needed 12 brains, so 12 aborted fetuses for one patient. So you can imagine how difficult it is to even coordinate this. And of course, it will be immune rejected. So originally the question was, can nuclear transplantation provide this? And as I said, it's not a real option. So the question is, would iPS cells be useful for this? So we differentiated in culture iPS cells to neural precursors. And these precursors can really differentiate glia cells and neurons in vitro as well as when you inject them into a mouse brain. And the question was, could there be in a Parkinson model function? The Parkinson model is the so-called Parkinson rat. And these rats are made by unilateral lesions by injecting 6-hydroxydopamine into one side of the brain, not the other. And that kills all the dopaminergic neurons on one side. I mean, the rats are okay after this, but if you inject them with amphetamine, you stimulate dopaminergic neurons, and since they're now unbalanced, right, on healthy side is normal neurons, the other ones doesn't have any, they begin to spin, they rotate. And the model is, if you now inject dopamine neurons into the lesion side, is there functional recovery? So do they rotate less, right? So you can see here, these lesioned rats, they're really 1,000 rotations in 90 minutes, so they really go, right? And you can see in this case, 4 or 5 injected rats really substantially reduced rotations. Actually, one rotated the other way. So clearly there were so many dopaminergic neurons that the lesion side was stronger than the other side. So at 8 weeks, 4 out of 4 operated rats had a major recovery. So what this argues really is not a real Parkinson model, but it argues these cells, iPS-derived cells, can function in vivo. That's what this really says. So the conclusion would be the therapeutic potential of reprogrammed virus is equivalent to that of yes, just for these two images. So when we think about what we want to achieve, actually, we want to take cells from a patient in a clinical setting and generate cells for customized therapy. So you take skin cells, get reprogrammed ES cells, and then cells are going to transplant. We're going to do everything in culture, of course. And I think the major progress here has been made. I think this is now more or less solved. There are technical issues to be resolved. I think here is a major issue to understand how to differentiate the cells you want to use to the right what you want to put in. So I think there are quite many questions, so I just address a few. I think the key question is how similar are iPS cells to ES cells? Very important question. I think we need a careful comparison to ES cells. And of course, we can ask the question, to what ES cell? There's so many different ES cells, they have so different properties. So I think it's very important to derive new ES cells under very defined conditions and figure out what is the best ES cell, what do we have to aim at when we try to make these iPS cells. I think there's much to be done. We want to know whether the reprogramming procedure really influences the properties. So, nuclear transfer versus iPS using different factors. And very interesting is an epigenetic memory of the donor cell type. So an iPS derived from a B cell, somewhat different from that derived from a skin cell or from a fibroblast. So I think that's again a very important question which we don't have an answer on. So on translation, of course for research, the oncogenes and the proviruses are not appropriate. We can do it today and it's being done. I think the key issue is, can we find a disease-relevant phenotype in the petri dish? And maybe for ALS, there's a good chance. For Parkinson, I think it will be a big challenge. We don't know if we find it. So that's really a big challenge. But for therapy, clearly, as I said, we have to figure out whether they have the same potential for therapy. We know, at least in the animal model, for sickle cell anemia and for Parkinson's model, that seems to be the case. The retrovirus oncogenes have to be eliminated. Nobody would be ever allowed in the FDA to use a genetically modified cell to put in a patient, so that has to be replaced by small molecules or by other means. I think a key question is what is the right cell for transplantation? It's not the ES cells or the iPS cells, they make cubes, so we have to be sure they're eliminated. But is it a committed stem cell? And how do we make committed stem cells? Or is it the terminally differentiated cell? Again, things we don't know at this point. And I think then delivery is probably the most important issue. So I think thinking that you could by transplantation therapy, treat a patient with Alzheimer is probably an illusion. You probably cannot do that. For ALS, might be very difficult. Parkinson, who knows, right? Maybe. But for blood diseases, because bone marrow transplantation is routine, that will be routine. Probably for diabetes 1 might be possible, because we know from clinical trials you can take beta cells from a cadaver, put them in a diabetes 1 patient. They function until they die, right? They function, and these patients become independent of insulin injections. So I think these are certainly diseases which probably will be more easy to adapt this therapy than to other diseases such as Alzheimer's or muscular dystrophy and so on. So let me just finish here and acknowledge the people who did this work. That was Jakob Hanner, Chris Lengner, Louis Bromberg were highly involved. The human experiments come from Maya Mitalikova, who's an expert in human ESS. She isolated 4 of the presidential lines with some, Frank Solomon and Kockemeyer, Stella Makulagi. Marius Wernick, who left my lab, is now at Stanford, was highly involved in all these experiments, as was Alex Meissner, who was his own lab at Harvard. Lauren Boyer, from a postdoc, is now at MIT, and he collaborated with Rick Young. And Tim Tones, who was a collaborator from the sickle cell, he made the sickle mice, and Ola Isaacson, at Leeds Hospital, his group worked on the Parkinson model, and also we got help from Martha Hansen and committee. Thank you.
Thank you so much. Very, very informative lecture. So this is a great opportunity to give everyone want to ask directly. Any question? Thank you very much for your excellent talks, Rudy. I have one question. So, did you see any change on telomerase length in your systems?
I missed your question.
Telomerase length.
Teratomas.
Oh, no. Telomerase.
Oh, telomeres. Oh, no, we haven't looked at it, but other people have. And like a nuclear transplantation, that was a big question first. Would Dolly be 6 years old or 6 years plus the age of the donor? And one original report suggested the telomeres were shorter, but this was then clearly disproved by many, many other studies in sheep, in cows, and in mice, showing that the telomeres are normal length. And this is to be expected, because telomerase gets expressed. And with iPS cells, there's some evidence, and it was shown in a meeting I just had been, that they're really telomeres are coming up to normal length. So Yamanaka actually showed that, I think, I remember. Yeah.
So that means there is a possibility that we can use this technique for the aging-related.
Sure, I mean, telomeres are not a problem. Axon activation is not a problem. So those things which occur after fertilization are not a problem.
Thank you very much. Any question?
Thank you for the interesting seminar. I belong to the allergy and immune research section, and I'm very interested in the immune cells therapy. You said that Hana succeeded in iPS or ES to B cell or T cell. Another cell succeeded information already exists or not?
So B cells to iPS cells, you mean? So they went from B cells to iPS cells, and then the iPS cells. Is that what you mean?
iPS cells to.
Oh, this would be
differentiate to.
Right, to erythrocytes. For the sickle cell anemia, you mean?
I want to know about the, for example, mast cells or macrophages,
yeah.
DC.
That is easy. That's more easy than lymphoid cells. Lymphoid cells in general is more difficult. But one should say that, so the mouse works well, but we use a virus, we use a HOXP4 virus. And of course, you don't want to do this for therapy. And so far, to make reproducibly, for example, adult cells, adult myeloid cells, which basically produce globin, erythropoietic cells, globin, from human ES cells has been very difficult. So I don't think that is resolved yet. I'm sure it will be resolved as well. Many laboratories are working on this very important.
Mast cells is very difficult to introduce it.
Because you get primitive, you get the primitive cells, only not the yolk sac derived type of cells, and not the ones which have the adult properties.
Thank you very much.
I have some. For me, it's slightly shocked. Disease model iPS cells not resemble, not showing degenerative disease state in vitro. So, what do you think? Yeah, you said a little bit about, but
I think that's the biggest challenge. I think. So what you could do is, I mean, there are various ways. What you could, for example, put on these neurons or on these cells, put oxidative stress, and ask the question: can you determine this, that maybe the disease ones are more sensitive than normal and healthy? Then you would have a system test, right? That's very complicated. So for some disease it might not work. I think one has to do an in vivo experiments. So one would have to make chimeras, I think, mouse chimeras, brain chimeras, and then having maybe a mouse which has human cells incorporated in the brain, and you just watch over the lifetime of the mouse, whether a disease-derived cell has a different phenotype than the normal one. If you do find this, at least you have a phenotype you can study. So, but this is really probably complex, and I think we haven't even learned how to do this yet. But I believe this will be one potential interesting approach.
So, I mean, just not only making disease model iPS cells, also we have to improve some cultural system or some.
Very much so. Yeah, I think that is probably the biggest challenge.
So, have you found any evidence of making iPS cells? You made a lot of iPS cells, and then terminally differentiate cell totally erased or partially erased.
So I think this is a very important question you're raising. So we have looked at this, but there's evidence from some people who talked at meetings. Konrad Loelinger's group was one, who show that in vitro differentiation of iPS cells to cardiomyocytes differs in iPS cells from that of ES cells, embryo-derived ES cells. That is comparing 5 with ES lines with 8 iPS lines. But the difference is 40% in one case to 60% in the other case. So I think it's a minor difference, and I find it's very difficult to quantitate in vitro differentiation. So I think it hasn't been published yet. But so that's an interesting issue. Is there a difference or not, and why? Is it due to the viruses? Maybe a little bit of expression of the viruses still, which are never really fully silenced. It's an open question, very important one.
Will that affect how much differentiation, or how to differentiate into target cells? What we can do?
Yeah.
It's not as similar as embryonic stem cells.
So I think the comparison between those is very important. I think there are many issues which need to be resolved.
Thank you for the impressive talk. I would like to ask something about B-cell-derived iPS cells. You mentioned that in the B-cell-derived iPS cell, the immunoglobulin has been rearranged already. So I wonder if the germline transmitted mice generated through iPS cell may have the immunological problem or not.
They totally have only one specificity, because it inhibits all further rearrangement. So they can only have this one antibody. They cannot, they're monoclonal.
Oh, so they have a problem.
Because the rearrangement inhibits any further rearrangement.
But
so
in terms of other differentiation, other cell differentiation in hepatogenesis?
Oh, that's fine. No problem.
Oh, interesting. In the slide you wrote like nuclear transfer ES cell / IP cell. Does that mean that NT ES cell and the IP cell are very similar, or is there some difference? Because from he did mention from his impression, NT ES cell resembles normal embryonic stem cell very well, but however the IP cell exhibits variation.
Yes.
So how's your impression?
So the first question was, of course, NT ES cells different from ES cells, and both Taro Wakayama we looked at this carefully. There was no difference. I mean, we come to the same conclusion. They were really identical by the criteria we can put on, biological as well as molecular. For iPS cells, from the mouse, from all the data we have, that's expression profiling, that is chromatin, that is methylation, they were really indistinguishable. There's one difference so far. One difference is tetraploid complementation. This is what I briefly described. So normally we have not been able yet to make a live mouse from iPS cells by tetraploid complementation. So there are two interpretations of this. One is they're really different. That's the most stringent test for herd immunity. It's much more stringent than germ-like transmission. That's really. So is there a difference, or we didn't try hard enough because it's a difficult technique? I don't know. And the other one is this in vitro differentiation, which Ida brought up to cardiomyocytes. So is there a difference or not? I think it's a question mark at this point. Not totally resolved.
From the viewpoint of the methylation status, I wonder if IP cell derived from many kinds of cell maintains its original phenotype.
That's a very important question, and we are very interested in some of this. We don't know.
Okay. So you also would like to ask about sickle cell anemia cell-based therapy, IP cell cell-based therapy. You are successful. You mentioned that you succeeded in treat the sickle cell anemia model and by injection of the IP cell. Is that the red blood cell generation of the red blood cell mediated through generation of hematopoietic stem cell or directly from the IP cell?
No.
Direct differentiation.
No, what we did is we differentiated those in culture to these stem cells, right? It was a pretty mixture, it was really, but there were enough stem cells which permanently can engraft. So they were really clearly multipotential hemolytic stem cells made in culture. I mean, we know this from before, but they're not pure. But all the cells which are the others get eliminated and die. So clearly it was engraftment, it was long-term engraftment.
My last question is that you also mentioned about the disease-specific IP cell. In that case, you showed several applications for the disease-specific IP cell. Could it be used? I may miss your talk, but did you mention that disease-specific IP cell can be used for the drug screening or the drug safety?
I think is a very important application.
Thank you very much.
Any question? Thank you. Thank you so much.
Gracias.